Good evening, everyone. Welcome to the Center for Evolution and Medicine's evening event, Precision Medicine 2.0. The times they are a-changing, and so is your health. My name's Ken Buteau. I'm the director of the Center for Evolution and Medicine, and I want to welcome everyone and thank you for coming this evening, spending part of your evening with us. I recognize that you all have many, many things that crowd your very important and busy lives, but we would like to hope that you will find tonight's event both interesting and engaging. And part of our goal for this session is actually to have participation from you all. What I'm excited about tonight is that tonight's essentially the community launch of the Center for Evolution and Medicine's Precision 2.0 medicine program. So what we want to do tonight is engage you, the larger ASU and or Greater Valley community, in this new exciting endeavor that we are launching. So I think it partially begs the question, what is precision medicine? So we're going to say that we're doing Precision Medicine 2.0. What is precision medicine? Well, it is the driving force, certainly in the biomedical community, for about the last 10 years that is intended to facilitate delivering to the right individual the right care at the right time and in the right place. Very aspirational goals that in many ways are transforming how we conceptualize medicine and public health. To date, though, most of precision medicine, the revolution that has become precision medicine, has been tied to the molecular revolution. Our ability and has leveraged the insights that we've been able to obtain from the sequencing of the human genome and the related technologies that allow us to routinely be able to characterize both an individual's genetic constitution, but also how their disease may be different at a molecular level. And now increasingly, not only just an individual, the human genome obviously about humans, but actually the true revolution of the Human Genome Project was the routine capability to be able to molecularly characterize our environment as well as ourselves. So we've learned of our microbiota. We've learned of the complexity of our immune systems. We've learned that the ecologies we live in are diverse and complex. So arguably, Precision Medicine 1.0 is about how we actually leverage our understanding of this molecular variation in driving health and evaluating disease risk, how we actually manage that molecular information. But what we're interested in doing and what we think evolutionary insight gives us the capacity to do is add a needed variable to this precision medicine in a Precision Medicine 2.0 universe where we actually not only capture the current or a snapshot of molecular variation, but how do things change over time? At its core, evolution is the study of how systems change over time, individuals and ecosystems. So in Precision Medicine 2.0, we bring into this the additional variability, complexity of sex differences, our full understanding of immune function, how we respond to our environment, what the environment is itself, how do these influence our behaviors, how do our behaviors influence these outcomes, and how do these all fit into the broader ecologic context that is required to maintain health and to understand disease. So this isn't easy. In fact, one of the things one recognizes is that in Precision Medicine 2.0, one of our fundamental pillars is the recognition that disease is complex. What do I mean by complex? It's different than complicated, because in complexity, what we understand is that the whole is more than the sum of the parts, and that we need to, in order to be able to understand the system, we can't just look at one piece in isolation and without understanding how it fits into a broader context. So A second pillar of Precision Medicine 2.0 is that context matters. Each individual and each system lives in a context of other pieces that in the absence of consideration, we don't have a full understanding of how that disease is manifest or how health is not maintained. We also recognize in a system of complexity that health is a compromise, that you can't manipulate one piece of the system without it having effects on others. And that in fact, mechanisms that produce health in one context actually can manifest disease in others. And we'll hear more about that as the evening goes on. And then lastly, that health is a continuum, that our changing ecologies, our life and individual experiences within a lifetime that faces actually change the pattern of disease for individuals and their environment. So we're very interested in actually embracing this complexity in our Precision Medicine 2.0 and our Center for Evolution in Medicine, and hopefully with your help, we'll be working to generate the evidence that evolutionary approaches in medicine and public health produce better health outcomes. That by embracing this, we can change the way medicine works and work to maintain health as well as treat disease. So as our center efforts, we actually are launching a series of pioneering Precision Medicine 2.0 signature projects. You'll hear some of the context of this tonight. We have 4 areas that we are concentrating transdisciplinary activities, embracing the new American University's point of view of the importance of transdisciplinary research activities, bringing together anthropologists, biologists, sociologists, psychologists, molecular biologists, all to surround and embrace the complexity of these problems and to treat them in context. So we're working with projects and more details in the packages you've received on how to understand sex differences in disease, studying the consequences of old adaptations in new environments, using evolution to solve drug and pesticide resistance, and exploring how ancient pathogens become modern epidemics. But one of the reasons we're having an event tonight is that we can't do this alone. In fact, we need you, the broader community of Arizona State University, the brain trust that is the Greater Valley of the Sun, as well as regional, state, and or national partnerships. So we're constantly on the lookout for who and how we can gain insight from others. So tonight's event is about engaging you, your feedback, your ideas. You're already committing your time, hopefully your talent, and maybe even your treasure, depending on what you're interested in and what you are capable of providing. So tonight's program, Precision Medicine 2.0, The Times They Are A-Changing, features 2 of our brightest stars in the Center for Evolution of Medicine's firmament, Doctors Ben Trumbull and Melissa Wilson. Ben will kick off this evening's event with an overview on the consequences of old adaptations in new environments, followed by Melissa, who will talk about sex differences in disease, and Ben and her's recent pregnancy pickle paper. Say that 3 times quickly. So with that, we'll hear a little bit about what the types of work are that we're undertaking, and hopefully, and then end the evening with a discussion and input, hopefully from you all, about ideas, ways to engage, and your reactions to the class of work. So with that said, I will turn the podium over to Ben and his work.
Alrighty, thank you all so much for coming tonight. So one of the challenges that we face as both a country and a world right now is that the US population is aging. So the Census Bureau expects that we're gonna have a 200% increase in individuals over the age of 85 by 2060. We used to have a population pyramid, but now we have a population column. Now, this is especially important for diseases like cardiovascular disease. #1 killer, 800,000 deaths per year. By 2030, it's expected to cost about $750 billion a year. Alzheimer's disease is the single most expensive disease an individual can get. By 2030, it's expected that there will be over $1 trillion in direct costs per year. But have these conditions always been a common problem? Has Alzheimer's and cardiovascular disease, have they always been major problems for humans? Well, one of the things is we live in a very weird environment at a very weird time. What we experience today is just the tiniest blip of the human evolutionary past. If you look at human population history, and you think about humans in general, for the vast majority of the human experience, we've been hunter-gatherers. So greater than 99% of the human evolutionary period, we were hunter-gatherers. If you were to imagine the last 5 million years of evolution, if you were to condense that into a single year, then on January 1st, we're hunter-gatherers, February 1st, March, April, May, June, all the way out until December 31st. And December 31st at 11:40 p.m., that's when the Industrial Revolution occurred. So the world we live in right now is just the tiniest little blip of the human existence. And yet we think of this as how our world is and how it shapes our health. The fact of the matter is, there's a really big mismatch in the environments in which humans evolved and the environments in which we live in today. So, I can get in my car and I can drive to McDonald's and I can hunt and gather 2,000 calories. Don't even have to get out of my car, no problem. I work with the Chimane, a group of forager horticulturalists in the Bolivian Amazon. If they want meat for dinner, they have to go for a hunt. Average Chimane hunt lasts 8.6 hours and covers over 11 miles. And even then, it only has a 60% chance of success. It's like walking 5.5 miles to the grocery store and realizing they're out of meat. So, this is a very different lifestyle. We have clean water, we have clean food, we have medication if we need it. But one of the things is, because all of the medical research that we do today is focused on individuals living our exact same life ways, we don't really know that much about what health was like throughout most of human history. So that's what we're going to talk about today. So, like I said, almost all of medical research is done in industrialized populations. So we're going to look at 3 deadly and costly conditions of aging. Heart disease, the #1 killer in America, benign prosthetic hyperplasia, an inevitable part of male aging in the US, and Alzheimer's disease. Now, much of this work is going to be situated among the Chimane. The Chimane are a group that practice a forager horticultural lifeway. So they get most of their calories from small-scale farming. They'll go out and clear an area of the forest, and then they'll plant crops like manioc root, corn, rice. They don't have electricity or running water, very little access to any sort of standardized medical care. Like I said, if they want meat, they either have to go fishing or have to go hunting. So it's a very different lifestyle than the one that we live in today. And you can't point to any single population of hunter-gatherers or forager horticulturalists anywhere in the world today and say, that's the perfect exemplar of the human past. Because humans evolved in a mosaic of environments. But where we didn't evolve was cities. And so we've never had things like clean water before. We've never had these ability to get into a car and drive anywhere and not have to worry about any sort of physical activity. So for a Chimane man to clear a field, he has to go out and first clear a whole area of the rainforest with just a metal-headed axe. And these are not small fields either. Then you have to burn it so that you can release all that carbon and other nutrients into the soil. And then you plant corn, manioc, plantains, these other sorts of materials. All meanwhile, you're out going hunting, and the animals that they're getting tend to be relatively lean. These aren't, fancy cattle that we grow in the U.S. that are, 50% fat. These are very lean animals or fish. Now, we know things like cardiovascular disease. Everyone knows that there are modifiable risk factors. So, my doctor says, I should go for more runs, I should watch my cholesterol, watch my diet. So we know that these sort of things can impact cardiovascular disease. And so a population like the Chimane, they've got relatively low lipids. But what about heart disease? Well, it turns out it's actually really difficult to diagnose heart disease in the US. So most people in the US don't actually know they have heart disease until they have a heart attack. There are some methods using nuclear cardiology or getting an angiogram, but those require a lot of effort. And so most people don't actually know if they have a cardiovascular disease until they have a heart attack. Well, one of the mottos of the group of individuals that I work with is that all methods are field-friendly if you want to know the answer bad enough. And so we sought out to go and see what is cardiovascular disease like in a subsistence population. Well, Chimane do have a slightly better diet than we do in the US. So about 14% of their calories come from fat versus about 34% of US calories. Now, one of the things that you'll notice, though, is that almost 75% of Chimane calories come from carbohydrates. So this is not Atkins' diet. And they get about the same amount of protein as we do in the US. Now, the cardiologists that I work with, they always tell their patients, get 10,000 steps per day, and that will help prevent cardiovascular disease. Unfortunately, the average person in the US only gets about 5,100 steps per day. The average Chimane across all age ranges gets over 16,600 steps per day. So a lot more physically active. But if you don't have a car and there are no roads to drive on, that makes it a lot harder to get around in a car. Well, like I said, it's hard to measure actual cardiovascular disease unless you had something like access to a computed tomography machine to actually use nuclear cardiology to look inside of a person. Well, fortunately, there are some really amazing methods using non-invasive coronary artery calcium CT scans. So this is a textbook image of an extreme blockage. So what you see here is this is a person, and you can imagine if you were looking down through the top of my head, that would be my heart there, and this white area here is my spine. Well, this isn't me, this is a textbook image. And that white image that you see inside of the heart tissue, that's actual calcification. So that means that this person has a major blockage and definitely needs a medical intervention. Otherwise, they're at a very high risk for a heart attack. Now, so if I'm working in the middle of the Bolivian Amazon and very remote communities with no electricity or running water, there aren't really that many CT machines hanging out. Luckily, we found a CT machine in Bolivia. It was donated by the French government, but it was broken, and they actually had no one trained how to use it. And so we brought down US cardiologists and US technicians from GE. They fixed the machine. We trained local hospital staff in how to use it. And then we started going through and systematically bringing people from remote communities, first to our local clinic in San Borja, and there we had a full medical workup done on those individuals. And then we called ahead to the hospital and set up all the surgeries and all the specialist appointments they need. Because you can imagine, if you're a 60-year-old Chimane and you've never seen a doctor in your life, you probably have some medical conditions that need to be treated, like your hernia, your cataracts, your pterygium, all of these things that you don't actually have access to or have the ability to fix yourself. And so what we do is we get them embedded in the local healthcare system. We bring people out on buses to the regional capital of Trinidad, where the CT machine exists, and then we go through and examine actual measures of coronary artery calcium. People stay on, get all of the surgeries and treatments they need, and then we return them back home. And so it's a win for everyone. Now, if you look in industrialized populations like in the US, what you see here is this is a coronary artery calcium score graph. If you have a coronary artery calcium score of 1, you have heart disease. If you have no coronary artery calcium, you don't have heart disease. So what this is going to show is if you look at the light blue, the second graph in, that is the median person in the US, so the median white male in the US. And that blue bar, what you'll see is that by the time you're age 75 to 85, the average US male has 400 units of calcium inside their heart. If you have 100 units of calcium, you should see a cardiologist every 6 months and you should be on major statins. This is not the case for the Chimane. The average Chimane male aged 75 to 85 has a coronary artery calcium score of 0. And it's not just for men, it's the same for women. And I should note that one of the lead co-authors on this paper is a SEM fellow, Dr. Angela Garcia. Now, for women in the US, the average US female has, by age 75 to 85, has a coronary calcium score of about 100, which again is significant enough that she should be seeing a cardiologist every 6 months. Again, not the case for the Chimane women. Only about 15% of Chimane had any coronary artery calcium, whereas about 85% of people in the US have coronary artery calcium. But more importantly, only about 2.8% of Chimane have actually high levels that are potentially dangerous, and they should be seeing a cardiologist or should be on a statin, whereas over 50% of people in the US in the same age range have very high levels of coronary artery calcium. And it's not just compared to the US. Chimane actually have the healthiest hearts in the world. So they have the lowest levels of coronary artery calcium compared to all of the populations ever examined, both for males and for females. Now, one of the things that we're doing now is we're expanding into another population living in the same environment, but living a much more acculturated lifestyle. And we're going to see if, given this same language group, very similar genetic structure, how living a much more acculturated life impacts cardiovascular disease. But one of the things that's really important about this is in the US, we assume that high levels of inflammation are causal in the pathway to coronary artery calcium or heart disease. And if you look at every aspect of a heart attack, from the beginning of plaque formation up through the actual myocardial infarction itself, inflammation is heavily involved in all of those processes. Now, Chimane actually have higher levels of inflammation than age-matched US individuals, but despite that, they still have these very low levels of coronary artery calcium. Also in the US, if you have low HDL or good cholesterol, doctors sometimes see that as a problem. And for the Chimane, they have extremely low levels of HDL, but still no heart disease. Now, like I said, most research is conducted in industrialized populations. A condition that's actually quite prevalent in industrialized populations is benign prostatic hyperplasia, or prostate growth. The prostate's a rare organ that just continues to grow with age. Now, prostate enlargement is considered inevitable in industrialized populations. After a certain age, doctors don't actually even ask their patients anymore because it's just assumed that they do have an enlarged prostate. It's considered an inevitable aspect of male aging. 90% of men in their 80s experience prostate enlargement in the US, and 40% of men will require treatment. If you look at disability-adjusted life years, despite only affecting males, benign prostatic hyperplasia causes more disability in the US than epilepsy and Parkinson's combined. So this is a very big problem. And when I say it's pretty much inevitable in industrialized populations, I really mean it. By the time you hit age 80 to 89, almost all males have an enlarged prostate. Well, what about the Chimane? So we know that there are three major factors that influence benign prostatic hyperplasia: androgen levels, obesity, and metabolic syndrome. And Chimane have relatively low androgens. They have very low levels of obesity and almost no metabolic syndrome. And so this could be a mismatch type situation where, because they're in a different environment and they have different metabolic and hormonal outcomes, they may have lower levels of benign prostatic hyperplasia. Now, again, our motto is that all methods are field-friendly if you want to know the answer badly enough. And so we did abdominal ultrasounds on about 350 Chimane men. It's much more pleasant than digital rectal exam, and we still wanted to work with this population. And so we felt that abdominal ultrasound was probably the best way to go. Now, if you look at prostate volume by age in other populations, so in the UK, German populations, US, you see a pretty steady increase in prostate size with age. One thing that I should note here is that a prostate volume of 20 cubic centimeters, that's actually considered a benign prostatic hyperplasia. So the average male by age 45 is already experiencing benign prostatic hyperplasia. This is not the case for the Chimane. Not only do they have relatively low levels, but they stay low across the lifespan. The average Chimane prostate is about 13 cubic centimeters smaller. They may not sound like very much, but that's about the size of a K-cup. And if you have something the size of a K-cup crushing your bladder and your urethra, it's kind of a big deal. On average, they're about 62% smaller, the Chimane prostate, compared to a US prostate. And importantly, advanced cases that would require, say, surgery in the US are almost non-existent. Whereas about 50% of males in their 70s in the US have prostates that are large enough that they qualify for medication, if not surgery. Now, while that's an important aspect of male aging, Alzheimer's is a very big problem that is going to be facing our nation very soon. So it's the most common form of dementia. It's a degenerative brain disease, and it's the single most expensive condition an individual can get. Unfortunately, its etiology is still poorly understood. It's increased in Arizona by somewhere between 45 and 75% in the last decade, and it's only expected to increase. Now, there is a strong genetic risk factor for Alzheimer's, and that's the Apolipoprotein E4 allele. It's the single strongest genetic risk factor, And individuals that are homozygous, that have 2 copies of this gene and are female, have approximately a 90% chance of developing Alzheimer's by age 80. Unfortunately, individuals who have this allele also have faster cognitive decline compared to non-carriers, and they absorb cholesterol faster and better, which for us isn't that great, because that means that if you eat a hamburger and you have the ApoE4 allele, you actually absorb more of that cholesterol than a person who was eating a hamburger and didn't have that allele. Now, while for us that seems terrible, you can imagine that there are some environments where that actually could be a little bit of benefit, getting to absorb a little bit more cholesterol faster. And there actually have been several populations now that have looked at children with ApoE4 allele. They grow faster and taller. They're also able to clear some parasitic infections faster. So almost all of the intestinal parasites that people face around the world, they all eat lipids. So they all eat fats out of either our bloodstream or out of the food we eat before we get the chance to eat it. And so if you can actually absorb your cholesterol faster and you're better able to clear those infections, that's actually quite an advantage. What you find among the Chimane is that they're able to maintain better cognitive performance despite very high parasite loads if they have the ApoE4 allele. And in fact, they have on average 1 to 3 children more if they have the ApoE4 allele. So what about Alzheimer's disease? Well, Alzheimer's is actually very difficult to diagnose in the US. It's a diagnosis of exclusion. So what you have to do is you have to first rule out B12 deficiency, traumatic brain injury, stroke, thyroid issues, depression, vascular dementia. You have to rule out all these other factors before you can diagnose a person with Alzheimer's disease. You have to do a bunch of different neuro exams, a bunch of brain imaging. And fortunately, because we have access to this CT machine, we have an ongoing collaborative project funded by NIH looking at how does cognitive function change with age among a population like the Chimane, and are they at the same risks for Alzheimer's that we are? And so just so you know that I do have a brain, what we're doing now is we've gone through and for almost 1,000 individuals, we've done all of the neuropsychiatric testing, we've done all of the brain imaging, and then we've taken all of that to USC, to their Alzheimer's Research Center there, and they're actually seen as typical patients. So we do the exact same interviews that they do, went and spent a couple weeks learning all of the different cognitive exams, and then they actually intersperse Chimane patients with their own patients when they do these consensus conferences to diagnose who has Alzheimer's and who doesn't. Now, we don't have results on that quite yet, but hopefully by next year we will be beginning to publish some of that. Now, at the end of the day, the problems that we face today have, in some cases, they're novel, like cardiovascular disease. That probably wasn't really an issue throughout most of human history. But we don't know until we look. See, human evolution occurred in a mosaic of environments. No one population is the perfect exemplar of the human past, but we know that humans didn't evolve in cities. Cities are such a novel, new environment, and they have so many different and odd challenges. And because most medical knowledge is gained from research in these industrialized populations, it's really hard to say what human health was like throughout most of human history. And so by understanding variation in human health and disease, we can better understand some of the causes and maybe some of the solutions to the problems that we're facing today and that we're going to be facing in the next 20 years. Thank you all so much, and I have the pleasure of introducing Dr. Melissa Wilson.
My lab hasn't traditionally studied pregnancy. I was a math major who then went to graduate school in bioinformatics, and I studied genetics, and I studied sex chromosomes. And one of the really neat things about being at Arizona State University is getting to be a part of the Center for Evolution and Medicine and getting to talk with people who think about things from very different viewpoints from myself. And so I'm going to introduce a little bit about what motivates my lab's research overall, and then I'll talk to you about a publication that we had called the Pregnancy Compensation Hypothesis, or the Pregnancy Pickle, which is something, it was a hill I was willing to die on for that name, and it's worth it. You can find me on Twitter, but I'm a part of the School of Life Sciences, the Biodesign Institute, and most relevant for tonight, the Center for Evolution and Medicine. And this is a X chromosome and a Y chromosome here. I'm not going to talk about them very much tonight, but they are my one true love. Okay, so what I want to also highlight is that when we talk about sex differences broadly, we can mean a lot of different things. We can be talking about the genetics, so we can talk about in mammals, or particularly humans, because we love ourselves, whether you have two X chromosomes or an X and a Y, or a single X chromosome with nothing else, or two X's and a Y. The complement of sex chromosomes that you can have can vary really tremendously. So sometimes when we're talking about sex differences, we mean how many and what kind of sex chromosomes do you have? One key point here, though, is that all of the genes involved in gonad and gamete determination are not on the X and the Y. In fact, many of them are spread out across the rest of our genome. So some of them are on X and Y, but sometimes we don't mean sex chromosomes, we actually mean gonads, right? That's what people are really asking you when you're asking you to check that box. Like, what kind of gonads do you have? And now it's going to be weird every time you go to the doctor. You're welcome. What are your gonads? What are your gametes? And what kind of gonadal hormones do you have? That's often what a lot of biologists are interested in. But those gonads don't always reflect the sex chromosome complement that you have. And then on top of that, we have societal expectations and different boxes for gender that people can fit into. And those don't map onto gonads or genetics one-to-one either. And that's, I think, for me, coming into biology, really cool. There's a lot of really fascinating things to look at the interplay between these. And largely, we've just been ignoring them for pretty much all of biomedical research, because for almost all of biomedical research, we've been studying European males. So we're getting a little bit better at it. In 2015, the National Institute of Health instituted a policy that says you need to consider sex as a biological variable. Doesn't mean that people actually are, but at least the NIH is on board with that. And it's one of the research trajectories that we have now, which is incorporating sex differences along these different axes into understanding how that's affecting human health. So if I can motivate us for a minute about human health, I thought we'd start with death, because we can only go up from there, right? So leading cause of death for men, anyone? Heart disease, yeah? Great, perfect. What about leading cause of death for women? It's also heart disease, right? Sometimes they get breast cancer, right? Cancer is, right, if you're not going to die of heart disease, thanks, Ben, number 2 will be cancer, right? So solid 50% of us are going to die of heart disease or cancer. But in both of those cases, men get heart disease or cancer at higher rates than women do. Or alternatively, women get these diseases at lower rates than men do. Number 3 is where we start to diverge. So women get stroke much more often than men. And there's a huge continuum here, too, in the risk factors for stroke. So we know that the immune system and even emotions can be important in that. So there's a bunch of interplay between stress and the immune system and stroke. And there's a kind of stroke called broken heart disease. And this occurs when someone has a stroke within 24 to 48 hours of a close loved one dying. And about 6% for cause of death for women. It doesn't show up till number 5 for men. It's unclear yet, though, whether this high rate of stroke in women has anything to do with the third leading cause of death for men, which is unintentional injuries. Shows up out of the top 5, about half the incidence for women. Number 4 for everybody is chronic lower respiratory disease. And number 5, the 5th leading cause of death for women is Alzheimer's disease. Shows up at number 8 for men at less than half the incidence. So for a while, the consensus was that we observed that women live longer than men. And if Alzheimer's is solely a disease of aging, then women should get Alzheimer's more often than men. But even when we age match this, we still observe that women get Alzheimer's at higher rates than men do. And work from people like Dr. Trumbull's group and others are showing that the immune system is playing a really key role here. So potentially we know the immune system's important in stroke and Alzheimer's disease, and that there are sex differences in these diseases. Fun fact, the X chromosome is enriched for genes expressed in the immune system. And we know that karyotypic women will have 2 copies of the X, and a karyotypic male will have 1 copy of the X. So this is what's typically observed. You'll notice I don't say normal, because I don't know which one of us is normal. but I know what's typical, right? So typically that's true, but the X chromosome is included in less than 33% of our genetic analyses. When I say included, I mean actually reported. We are, earlier this week, I realized there is a name-redacted major data set that produced just a ton of data, and you can download it, you can access, you can look at gene expression across 1,000 individuals, and they just didn't report the X at all. It's just absent completely. So it's frustrating to me that we recognize that there are tremendous sex differences in incidence and disease, and we're ignoring 5% of the genome that could account for some of these sex differences. Okay, so how'd that get me here? Well, it's a long and fun story. But what I want to talk about is the pregnancy pickle. So this is the work of actually 2 SEM postdoctoral fellows, Dr. Haney Natry and Dr. Angela Garcia, and then Dr. Ben Trumbull and Dr. Ken Buteau, and they let me lead that circus for a little while. And it was really a fun experience because I don't think in the same, I would say all of us think in very different ways. So it was really a wonderful example of how people who think about subsistence populations and health inequities and genetics and cancer and genomics all kind of come together to come up with something. So we're basing this hypothesis first on the observation that there are these tremendous sex differences in disease. Well, why? If we could figure out why, then maybe we can do a better job about addressing the sex differences and understanding the mechanisms of the disease. Okay, so to start out, we know, as Dr. Trumbull said, there's a mismatch between ancestral and modern environments, and that includes the modern immune environment. And so on the y-axis is immune modulation. This is a sort of hand-wavy example because the immune system's pretty complicated. There's T cells and B cells and interactions of a lot of different components of the immune system, but we know in ancestral environments, there was no sanitation, there was no access to clean water, there was a tremendous amount of pathogens and parasites, and so people were constantly exposed to this high pathogen load, and over those millions, and in fact, hundreds of millions of years that vertebrates and mammals evolved, they were constantly exposed to parasites and pathogens. So your immune system evolved to respond to that. So we should have the immune system pushing back against that high parasite and pathogen load. And then with industrialization, we just have suddenly, in this very, very little blip, a low pathogen load. So our immune system is primed to respond to this high pathogen load, but it's suddenly not there anymore. And so we observe incidence of autoimmune disease across all individuals in industrialized populations at higher rates than has been observed in subsistence populations. And to my knowledge, there hasn't been a report yet of an autoimmune disease in a subsistence population. But we have much higher rates of autoimmune disease and also cancer in industrialized populations. Well, you told me this is about sex differences, and I'll say, oh, well, actually, there are quite a lot of sex differences between what we anticipate to be the ancestral environment and the industrialized environment, and that's that women experience autoimmune diseases at rates as high as 8 to 10 times higher than men. So in rheumatoid arthritis, it's about 8 times more frequent in women than men. But also I showed you that women get cancer at rates that are lower than men for almost all non-reproductive cancers. So one of the ideas is that there might be this interplay between having a higher immune response for this incidence of autoimmune disease in women, and maybe that higher immune response leading to higher immune surveillance and reducing or maybe catching some of the cancer a little faster. Why, right? That's what we'd like to know is why. And so what we're proposing is that there's another big difference. So there's a tremendous difference in pathogen load between our ancestral and industrialized environment, but there's also a tremendous difference in reproductive ecology. So prior to modern urban industrialization, women were pregnant or could be for the entirety of their reproductive career. There was no real way to stop it. We didn't have contraception. And so this little peaks are saying that the immune system is kind of bouncing back and forth. We know that the immune system in women who are pregnant is downregulated because that individual who is pregnant has to tolerate a genetically distinct individual. And so we know that the immune system bounces back and forth when people are pregnant or when they're not. And we know that people with autoimmune disease, some autoimmune diseases, so in rheumatoid arthritis, about 75% of women with rheumatoid arthritis, their symptoms go away when they're pregnant. They completely ameliorate. It's the best treatment that has been identified for rheumatoid arthritis. But we know in modern industrialized societies, we have much lower parity, right? And here's the point where I will note, right? What some people would like to do is then say, yes, well then, obviously, if the ancestral state was being pregnant all of the time, then modern women should be pregnant all of the time. Great. Also, give me back your sanitation. Give me back all your antibiotics. Give me back your vaccines. You don't need clean running water, do you? Right? No. Just because of how something was doesn't mean that's how it should be. That's called the naturalistic fallacy, and we don't have to succumb to it. What we do need to acknowledge is that how it was shaped our immune system, and it shaped our bodies overall. It doesn't mean that's how we should be now. Don't go eat handfuls of dirt. Please wash your hands after you go to the bathroom. I'd appreciate it. So maybe we'll take a little step backwards. How do we think we evolved these big sex differences? We're going to take it all the way back to mammals. So these are the mammals that were all sequenced at the time that I did my PhD, including my favorite mammal in the corner there. She's 8 now. Oh my gosh. One of the things, just to put in context, so this is a phylogenetic tree which shows the relationship between species. So eutherian mammals, which are sometimes called placental mammals, but I will tell you why that's not correct in a minute, are most closely related to the marsupials. These are the pouched mammals that we find in Australia, and there's that North American marsupial, and then the monotremes, and then those are all mammals, and then we have birds. And so mammals have been around for about 170, 200 million years. What's really interesting, though, is that we've evolved different ways of reproducing. So birds have these hard-shelled eggs. The monotreme mammals, so the platypus and echidna, have these kind of leathery, soft eggs. Marsupials form a very proto-placenta, but then they birth this little jelly bean, and it crawls up into the pouch and attaches to the nipple for several months. If you haven't seen a marsupial birth, you should YouTube it later. It's really, really fascinating. And it's a lot less traumatic than a eutherian birth, which we have a fully formed placenta. It's a little bit over the words there, but we have this, I say fully formed and invasive placenta, because the placenta in humans and primates specifically, and we think ancestrally, was hemachorial. So that means that the placenta itself, which is derived from the developing embryo, not the mom. The placenta is not the genotype of the mom. The placenta is the genotype of the offspring. It's growing. It's helping provide a barrier or interaction between the maternal body and the developing embryo, but it's also blubbing off these signals to downregulate the maternal immune system. It interfaces. It actually forms these tendrils up into the maternal body. The maternal body has to form a second tissue called the decidua, which we never talk about, that pushes back against the placenta. It's like, hey, baby, yeah, I super love you, but also don't take all of my nutrients away. And so we have this very, very different form of development that marsupials, monotremes, and birds don't have. Okay, now the nerd out for just a minute. A thing that has bothered me for my entire career is that also at that same time, our sex chromosomes switched. So the sex chromosomes, there's this X conserved region, Y conserved region. It's an old XY. The XY that we have is not shared across all mammals. In fact, that's not a genetic feature that's a mammalian feature. The monotremes, the platypus, has 10 sex chromosomes. They couldn't figure out what to do. It's just like, it's wild and interesting and just kind of fitting, right? But marsupials and eutherians, we have this conserved X and Y. So at the very first instance of having internal development, we developed a shared XY system. And then in the common ancestor of all eutherian mammals, all of the mammals with a fully formed invasive placenta, we added a second chunk of DNA that's 50 million bases. It's a third of our sex chromosomes that we added to our sex chromosomes at the same time. And to me, this has always been wildly fascinating because there's a bunch of genes in there that are expressed in the immune system and may have played a role in allowing invasive placentation and the kind of internal development that we have now to occur. So it's one of the things that my lab is continuing to investigate. On top of that, so I won't go into this too much, there are very many differences between traditional human environments in these subsistence populations and in sedentary industrialized populations. So we've talked about the parasite load being very different, parity or number of reproductive, or the reproductive ecology being very different, numbers of calories being very different, and also levels of reproductive hormones are quite different. And so all of these can interplay together to affect sex differences in disease. So we're hoping to kind of come together and say, we think we can explain sex differences in disease using a series of different evolutionary and proximate mechanisms. So we think that functionally, this is because of the immune system. We think the immune system is functioning differently in typical women and men. Well, how does that happen? Why did that happen? We think that this was largely driven by evolution of the placenta and the internal development and gestation and the interaction between that genetically distinct individual. For all of the rest of mammal history, if something invaded your body, a bacteria or a virus, it shouldn't be there. It's genetically distinct. Get rid of it. But if you get rid of that genetically distinct individual, you will no longer have offspring and the entire species will go dead. Thankfully, we haven't yet. We figured out a way around it. And how did we figure out that way around it? Well, we think through a combination of reproductive hormones, regulating the immune system, and dosage on the sex chromosomes. So we have these chromosomes that occur in 2 copies in half the population and 1 copy in the other, and it's a way that you could directly modulate reproductive hormones. And then the last is exacerbating this. The move from being non-industrialized to industrialized populations changes the overall levels of reproductive hormones. So whatever they were to modulate the immune system, now we're just being blasted with it. Reproductive hormones are much higher in industrialized populations across the board than they are in non-industrialized populations. So my lab is studying this in a variety of different ways. We're looking in all collaborative projects, studying sex differences in liver cancer, sex differences in the placenta, because the placenta itself will be XX or XY. And we think that sex differences in the placenta itself might be leading to differences in health outcomes for births. Sex differences in the brain, sex differences in human populations, and broadly across the immune system. And so with that, I don't want to take up too much more time. We're really happy to be funded by the National Institute of Health and supported by the Center for Evolution Medicine and the School of Life Sciences. And thank you all. And I think I will leave this up for questions so we can have discussion.
So we'd like to open up to questions, comments, reflections from the audience. I think we have microphones here that we can actually share for if people have questions. And I guess while people are stoking up their courage, I at least, oh, please, okay. Yeah. No, good, actually.
So you just ended a topic about our sex differences and our hormones, and you ended by stating that in the industrialized communities that our level of exposure to hormones is much greater. Can you elaborate?
Oh, I could, but I might pass it off to Ben because that's his area of expertise.
Yeah. So thank you so much, Dr. Wilson. One of the things that's really interesting is that if you are in an industrialized population, you're going to have far fewer kids than a subsistence population that's natural fertility. So the Chimane that I talked about, the average woman has 9.1 kids. And that means that almost all of her adult reproductive life, she's either pregnant or she's lactating. And so there's been some really good work on another subsistence population that has about 10, that averages about 10 children per woman. And in that population, they average about 109 menstrual cycles across their entire life because they spend most of their time either pregnant or lactating. In the US, the average US woman has about 450 menstrual cycles. What that means is that women in the US are getting exposure to 4 times the hormone levels that you'd see in an industrial population, just because you get all this additional estrogen, all these additional unopposed days of estrogen, where you have high levels of estrogen just before ovulation, and then there's no progesterone at the same time, because estrogen is actually a carcinogen. Now, the same thing's actually true for testosterone. So testosterone is also a carcinogen, and guys in the US have about 3 times higher levels of testosterone than age-matched males in subsistence populations. And part of that's because testosterone and estrogen are both expensive to maintain. So testosterone codes for muscle tissue. And muscle tissue in males, that's about 20% of the calories that I eat per day just go to maintain my muscle mass, not even to add any more. And so one of the things that you see is that if there's any sort of issue with the number of calories coming in, so if I fasted, if I just skipped dinner, tomorrow morning I'll have 10% lower levels of testosterone than average. If I fast for 2 days, I'll have castrate, or basically 0 levels of testosterone. During U.S. Army Ranger training, so very healthy young males, they have a intensive skills week, and during that skills week, they're burning about 6,000 calories per day, and they're often eating less than 1,000. By day 4, castrate levels of testosterone. And so because we live in an environment where we have abundance of calories, and because we aren't having kids all the time, we actually end up with relatively high levels of a bunch of different hormones. So if you look at breast cancer rates in subsistence populations, they're about 100-fold lower because they have such lower levels of estrogen exposure and relatively low levels of estrogen to begin with.
Please.
Yeah.
Tell me to stop if you guys want to talk. So you can't have this conversation without the blaring, obvious manipulation of our society with exogenous hormones.
So, interestingly, back in the 80s and 90s, there was a big push for hormone replacement therapy for oh.
It's everywhere since we started using pills in high doses. Oh, sorry. Right? Because that's our biggest environmental influence.
Well, yeah, so one of the things is that most of the hormonal contraceptives that are used today are progesterone only. And so there aren't any estrogen only. Just because after the Women's Health Initiative study in
Women Health Initiative came out probably, like I want to say 2005 for, that's only pertaining to hormone replacement therapy for the perimenopausal, postmenopausal woman. So if you look at the dosing of estrogen, since estrogen is the primary carcinogen, it's 1/10 to 1/50th the dose of OCPs that were introduced into our population in the 1950s. And our OCPs continue to go down in concentration, but they're still 10 times greater than what was studied in the Women's Health Initiative. So most women stopped, right? Postmenopausal, perimenopausal women, we were told it's bad. But at the same time that was happening, we were promoting artificial contraception to prevent unplanned pregnancy and to treat menstrual irregularities and to treat mood disturbances like PMS or painful periods, because it became the blanket answer. So is anyone looking at societies that are not doing that and looking at those same levels of disease?
Yeah, so one of the things that's interesting about that is that a lot of the, so if you look at hormone levels in Bangladesh, so in Bangladeshi women living in a low-income setting, they actually have 50 times lower levels of progesterone compared to an age-matched US woman. And so if you take the hormonal contraceptives that are oftentimes used in industrialized populations, and so big missionary groups will come together, or other really well-meaning NGOs will collect a whole bunch of US-based hormonal contraceptives and then donate them in these other populations where individuals aren't exposed to those same hormone levels. And then you see all sorts of side effects from that, because suddenly people are getting blasted with incredibly high levels of hormones compared to what they're used to. And these are all well-intentioned individuals. They aren't NGOs out to harm people, but it causes all these issues. And one of the things that you see today for testosterone replacement therapy. If you watch a football game, every third ad is like, do you feel tired? Like, do you have low energy? It must be testosterone. And so all of those men are basically entering themselves into a big clinical trial, and they don't know it yet. So they're going on these relatively high-dose testosterone patches or pills or creams, and what we're going to have, once the Men's Health Initiative comes out in 10 or 15 years, once we have enough data, it's going to be the same sort of thing. We're going to realize that all of those guys are putting themselves at a very increased risk for prostate cancer or benign prostatic hyperplasia.
Other questions or comments or observations?
Thanks very much, you all. Actually, I was wondering now when I was listening to both of you, is that often the comparison is made between Western populations, right? Well, not even Western, US populations compared to modern hunter-gatherer societies, right? And then you're talking about parity, for instance, but we're also talking about diet. So there's in city life, so there's a lot of things that are different. And I was wondering, is there also a lot of comparisons then on non-US but Western lifestyle populations? So I'm thinking of Mediterranean lifestyle populations that have a healthier diet, but they still have the same issue of living in a city. They have very low parity. So what would the comparison be like then?
Yeah, I mean, there are a lot of studies, especially in the Netherlands, or not Netherlands, sorry, you are from, in Denmark. There's a lot of studies in Denmark where the diet's very different. I don't know about Mediterranean diet, but in Denmark, where people are more active than in the United States, you still see a lot of sex differences in the diseases. So I'd say there's quite a few studies in Europe, but we're missing a lot in East and South Asia, and that's actually Dr. Buteau is working on that. But even some of the sex differences observed in cancer are actually magnified in some of these populations. So trying to understand the relative role of hormones and genetics is something that we're trying to grasp at, because we'd like to know which components are things that are more modifiable and which are others that are general risk factors.
Yeah, and just to comment to what Dr. Wilson is referring to is, like, for instance, we're beginning to try to undertake studies in India where you clearly have industrialization, you have urban environments, but the diet is very different. Nutritionally, things are very different, actually, and much of the other lifestyle variables are very different. The disease patterns are complex. They are scarily and very rapidly having the diseases of formal westernization at increasingly crazy rates. Actually, arguably, and there's some interesting mechanistic hypotheses about this, that actually at rates higher than people that have had generations of exposures to these Western diets and more westernization. And there's some questions whether that's based on other genetic backgrounds, whether it's based on epigenetic differences that have to do with that are intergenerational imprinting class differences that make you, when you suddenly have a McDonald's on every corner, when you've transitioned in a single generation to actually having McDonald's on every corner, as to oppose the more gradualist that's happened in Europe and in the United States, that maybe there are direct effects of that. But interestingly, it's intriguing to me how understudied these things are. And given our lack of success in addressing many of the diseases of westernization, we still, I think, do a really crummy job of really understanding the etiology or how to prevent or manage health around cardiovascular disease. Cancer's been a debacle, in my humble opinion, with respect to actually having any impact in real cancer prevention, short of removing tobacco exposure. But for the rest of the cancers, we really have almost no idea how to mitigate risk for those in a preventive context. So I think there's just tons of opportunities in that space.
So to also bring the hormones back in, the one thing I do know is that we're studying breast cancer, and you don't often associate androgen receptor with breast cancer, but androgen receptors are healthily expressed in breast tissue. And in fact, in most hormone receptor positive breast cancers, so estrogen receptor, HER2 receptor, progesterone receptor, you have androgen receptor expression. In triple negative, so triple negative is the kind that doesn't have estrogen, doesn't have progesterone, and doesn't have HER2, so triple negative. It's the most aggressive, it's most difficult to treat. We're finding that there might be a quadruple negative that also doesn't have androgen receptor expressed. Triple negative is, in the United States, the most rare kind, but it is in fact the most common kind in China. And so we'd have no idea why that is. Why? Maybe it's exposure to estrogens in the environment as well as not in the United States that's leading to that difference, or there could be some other environmental factors in China that are leading to a much higher, or in fact the highest incidence of triple negative.
And a real-time data point, triple negative is the most common form of breast cancer in India.
Really?
It's much, much more common in India than it is in the United States.
Well, and that's terrifying because we can't treat it. We just do terrible with it compared to the others. And so, yeah, I think trying to bring in some populations that have varying levels of industrialization and diet would be really, really great.
Please.
I love how polite everyone is.
Great.
I'm just curious, given the fact that X and Y are often left out of our genomic studies, which is disappointing. I'm wondering if people are being a little more conscious of the use of analyzing those data, and are they being included in the UK Biobank? The US is trying to do all of us, which is somewhat, we'll see how that goes. But, meanwhile, Iceland has sequenced half the population, and Finland's done 10%, and I forget what Denmark's at, and the UK's got, I think, 10,000 genomes. So are they beginning to kind of address some of those gaps with those kinds of data?
The short answer is no. So just a couple weeks ago, I was at a meeting about interoperability. So how do these big data sets talk to each other? And the one thing that none of them do is appropriately address X and Y. And there was a big discussion about, well, do we try to go back and reprocess everything? Because it's not so, if you failed to address it appropriately at the beginning, you actually have to just start over functionally, because we take a tissue sample and we crush it up, and it's not like X and Y are just hanging out by themselves. All those DNA reads are everywhere, and you have to realign everything. And the projects were really kind of pushing back and forth, saying, is it worth including them now? Is it worth just saying, well, we've already, this project's 10 years old. Let's just leave it to someone else to do. Amazon's going to be big enough that someone can, whip out $15,000 and redo it. It was interesting that across the data sets, I'll not point fingers at anyone negatively, but very positively, the Kids First study. So there's a study about childhood disease, and they very critically were really interested in incorporating sex chromosomes now and saying, it's worth it to us because even prior to having reproductive hormones at puberty, you see young boys get most all cancers at higher rates than young girls do. So pediatric cancers have a sex difference also. So it's a really clear case where we see strongly a genetic component to it, and they just hadn't taken the time to think about how you would include it, but they were very, very on board with doing it. So I hope that will, if we have a few small voices shouting, eventually we'll get it into traditional pipelines. Like if we could get it incorporated into GATK with the genome analysis toolkit, I think that would help a lot.
And just to amplify that point a little bit is, I think your point is well taken that the data is being generated. It's part of a natural process of actually how we do our genetics and genomics molecular evaluations. But Melissa and I just did a paper, and we're inventorying the number of papers that were actually even mentioning sex chromosomes in these large-scale genomic analysis in 2019. I mean, this isn't something that we go back and, well, it's the sins of the past. This is in 2019 publications coming out. The number, it was like single-digit percentages of the genomics papers that were still that we're incorporating it. And I know there's a air of apology that says, well, all of these original resources were underpowered to be able to look at this, so that's why we don't do it. But at a certain sense, I mean, that's an excuse. I mean, because what we've found, intriguingly, and I guess maybe there just needs to be some high visibility publications, is that these effects are not only there, but even in the underpowered studies, they are screamingly large. And in fact, when you do the sex segregated analysis, you find signals that were dampened because you were averaging males and females, and they were canceling each other out. So we find more signals when we do sex segregated analysis than we find in the combined double sample size that you would get for the sex averaged analysis. So, other questions, comments? We're happy. No, please.
Yeah. Also
Sure.
pitch that later this semester, the first week of December, Chris Katsapas is going to be here, who studies all autoimmune diseases and trying to figure out. He's actually doing some really cool immunological work in cell lines to figure out the relative role of hormones and genetics on incidence of autoimmune disease. So he'll be here. You should go to the SEM website and sign up. Everybody can come. You can meet with the speaker afterwards. So, yeah.
So this is a bit of a remediation question. You mentioned the Alzheimer incidence and the ApoE4, and I don't truly remember, but there's a buildup of plaque with people who have the ApoE4. And you suggested that in your Amazon population that they have the ApoE4, but because they have a high parasitic load, they're going to have a different incidence of Alzheimer's. Are you looking at what is activated by the parasite in the neurochemistry? Because we didn't talk about that, and that sounds fascinating.
Yeah. So there's actually a really interesting new hypothesis about Alzheimer's disease. So technically speaking, you can't diagnose with someone with Alzheimer's truly until after they're dead. And then if you do a section of the brain and they have amyloid beta plaques, then that person had Alzheimer's. And so what you have to do is you have to do these consensus conferences where a team of like neurogerontologists, neuropsychiatrists, neurologists all review the case. They all review the brain CT, or usually MRI in the US, and then all of the different tests to rule everything out. And then they make a consensus diagnosis. Well, there's this interesting team at Harvard, and they were studying amyloid beta, and they realized that the amyloid beta peptide itself actually looks really functionally similar to a really potent antimicrobial. And so I said, well, let's just try this out. So they took amyloid beta, they put it on a petri dish, and they seeded it with bacteria. And it turned out that bacteria wouldn't grow on it. So then they took a bunch of Alzheimer's brains and a bunch of control brains, and they ground them up like you do normally. And they put them onto petri dishes, and then they seeded them with bacteria. And they found that the control brains grew bacteria just great. But these brains that were from Alzheimer's patients grew at a much lower rate. And then they looked within those Alzheimer's brains, and they found that the samples that had higher levels of amyloid beta, like more amyloid beta per gram, actually had poorer growth. And so they started to come up with this idea that what if amyloid beta is actually part of the brain's immune system? Because we don't want a bunch of white blood cells up there, because they cause way too much collateral damage, and we would not be functional. And so what they did then was they used a mouse model. And mouse models for Alzheimer's aren't great, and so keep that in mind. But what they did was they took normal mice and then these Alzheimer's mice, and they injected them with really high doses of radiolabeled pathogens. And as we age, our blood-brain barrier that normally prevents pathogens from, say, getting into our brain, it starts to break down and become leaky, and some of those pathogens can make it across. And what they found was that the control mice mostly died, and they died relatively quickly. The Alzheimer's-type mice lasted longer, and some of them actually even survived the full 5 days before they sacrificed them. Then what they did was they looked inside their brains, And they found that in the control mice, a lot of them died of meningitis. So pathogens got into the brain, and then they died. In the Alzheimer's mice, they actually found amyloid beta plaques that formed around those radiolabeled pathogens and walled them off and killed them. And so we actually, we are still in the very early stages of data collection for working with the Chimane, but one of the possibilities is we're actually going to see higher rates of Alzheimer's because they're exposed to more parasites and pathogens. So the more parasites and pathogens you're exposed to, the higher probability that something is going to make it across the blood-brain barrier. And then if something makes it across the blood-brain barrier and amyloid beta plaques form around it, then that's the beginning of Alzheimer's. And so if you're in a much higher parasite and pathogen load, then we may actually see higher rates of Alzheimer's type dementia. We expect to see much lower rates of vascular dementia because we know that they have such low levels of coronary artery calcium. And so the atherosclerotic processes that cause that to form in the heart are the same that cause the carotids to form. And so we're not expecting to see much vascular type dementia, which you see a fair amount of in the US. But we may in fact see more Alzheimer's type dementia, but we just don't have the data yet. We're getting close though.
Please.
If you'd speak more about your overall goals for the 2.0 initiative, and in particular trying to demonstrate that this kind of information is going to lead to better clinical decisions.
Mm-hmm. So part of this, and this is part of the Bearing Trust that's actually putting this together, is the idea is that by, for instance, what Ben was just suggesting, for instance, with respect to better understanding the evolutionary dynamics that might be driving amyloid, beta amyloids, or that we actually will be able then to ultimately have a whole different set of diagnostics that will allow us to be able to find disease earlier, or conceivably interventions that would help us modulate disease actually through maintaining health as opposed to actually treating disease. So really embracing both the public health and medicine paradigm by better understanding the etiology of disease, we can actually intervene in it earlier and differently than actually trying to treat the end stages of disease. So that's the broader philosophy of that. And these individual projects are all then literally targeted at finding, well, first understanding our mechanisms so that we can have better biomarkers of susceptibility and that we can then be monitoring disease course and disease progress. And hopefully some of these biomarkers will then ultimately be different targets for intervention. So in the pregnancy compensation hypothesis, it's been almost completely unexplored, for instance, in cancer, how the female immune system is different than the male immune system. For a variety of both commercial and practical reasons, when we do immunotherapy in cancer, we treat women and men as if there's literally no difference in their immune profile. Well, if we can better understand why women actually have, in certain cancers, a 4-fold less likelihood of developing cancer than men, we actually know that half of the population can tolerate whatever that is that actually reduces cancer 4-fold in women. If we could actually, to be honest, bottle that, it could be actually transformative in how we would actually prevent and treat cancer. So that's the kinds of ways that we're seeing how it would change practice.
Are you making any efforts to specifically plan to get particular types of goals?
Oh yeah, no. So we, and one of the things I'm happy to share, but it's difficult in this, we have a whole strategic framework by which we're working at to actually develop these programs, working with local area communities. So I work, and I know Dr. Sayers works with, or Dr. Wilson works with the Mayo Clinic. We are working with a new group that called Equality Health that we're actually exploring a partnership with. So we're working directly with the clinical communities within the Valley to actually be doing these studies in real time. So
The group at the Mayo Clinic, there's a variety of different cancers, but we have a paper in review right now looking at sex differences in glioblastoma, which is really notable for Arizona and Senator McCain and others, which affects men at much higher rates than women. And many of the drugs have failed with treating glioblastoma for men, but it turns out, so Joshua Rubin's lab, who collaborates with the Mayo Clinic, he's at WashU in St. Louis, has been going back and testing those in XX cell lines. Because it turns out not only do we study these drugs in males in clinical trials, almost all of the cell lines are XY also. And he's been able to recover some of those as potential treatments for women because they failed in men, but they seem to actually be working in women. So roughly 10 to 20% of them. So I think by taking some of these viewpoints, we can even salvage some of the things that have failed clinical trials already. Yeah, right?
Cool. Anything else? We can hang out a little bit afterwards, but I'm sensitive to people's time, and I appreciate for everyone's participation tonight. And thank you for attending, and look for us. We will certainly be tweeting out and actually sharing and emailing all of you for future events, and hope to see you at a future Center for Evolution and Medicine event. So thank you very much for your time tonight.